So welcome everyone. It's a pleasure to see you all here in this joint webinar between LABI and LIBRE Hub. So, and thank you very much, Tobias and LIBRE Hub, for this initiative. That I think that it's great to have it. And now I'm going to pass the microphone to Tobias, that's going to present this webinar. Thank you very much.
Thank you, Oliver, and thank you, Katy, for joining us today. So it's really exciting to have you. I actually had to make some notes because I rarely introduce people that are so distinguished. So I need to list a couple of important things just to give you an idea why I'm excited to have Katy with us today. So Katy Börner is, well, originally German, actually, so we share that, but we're not living in our country anymore. And she is the Victor H. Juve, actually, I'm not sure how to pronounce that, Distinguished Professor of Engineering and Information Science at Indiana University, and founding director of the Cyber Infrastructure for Network Science Center. And she's involved in quite a lot of important initiatives. So I definitely don't list them completely, but you will see a number of them in the talk. An important one is the Human Reference Atlas, for example, where she leads the initiative to create sort of a common coordinate framework for shared vocabulary, to see what we're actually talking about and to visualize biology in 3D. And one of the things that brought us here actually in a discussion, even though it's not so featured now in the description of the seminar, is that she authored a new book that's called Atlas of Macroscopes, which is a very, very cool collection of really large data sets that are visualized in interactive ways. And I explored several of them, and they're very broad, not just biological. And it's really, really fascinating to see how information can be presented and visualized, and through that you gain interesting understanding and things that I had no idea about before. So that's really exciting. And she's also the long-running coordinator of different sort of science communication outreach initiatives like Places and Spaces to map sort of science and data visualization maps and more generally. And yeah, and she just has this mission that I think is very relevant for us in LABI about making scientific information more connected and accessible. So I'm really excited to have her here with us today in this community and to hear from her different initiatives and examples. I think there's a lot, she could give a lot of very different talks, I think. So now there's going to be some examples on, focusing on biological images, which is an important part of the work. But there will be a lot of links that I will also take from the slides and put into the chat throughout the seminar so that you can then also explore the other examples in your own time. Welcome, Katy.
Thank you so much for the kind introduction. It's really nice to be here. I was hoping to do this all in person. We recently had a CIFAR Multiscale Human Atlas project meeting in, near to Santiago, Chile. And I was so scheduled to go there and to go to Chile for the very first time, and then to also explore some of the wonderful mountains you have there. But then I had something happen in my life where I couldn't come any longer. And so I really found that super sad. But then Tobias found a way to get me to connect virtually. So I'm really grateful for that. So the presentation I have prepared is on the Human Reference Atlas, because I think that really connects us. We do a lot of spatial data within HubMap and also within CIFAR, and you have some of the links in the chat. But then also leading over to the data visualizations that we have been curating and creating and building tools for, so that more and more people can render data into actionable knowledge. So it's good to take data and analyze and model it and make it understandable to many. But if you just understand it, but you can't act on it, that's not as valuable as if it really fits into your daily workflow, if you can really then make better decisions locally, but also globally, short term, but also long term. And I think that's especially right now important, as we have some forces on this planet which are very local and very short term. So let me share my screen here, and hopefully you can see it. So you should see the title slide with humanatlas.io in the line. And then I will just go through the slides. And thanks again for having me. I also tried to repost on our social media. This way you get to see what else we are doing. It's a really interesting international collaboration that is creating a community resource available for anyone to use. All data, all code is licensed in a way that you can use it in teaching, in research, even in startups. So we want that because ultimately somebody has to provide 24/7 services. And of course, we want this data to be used. It's really an effort across many different consortia. You see some of them listed here on the left-hand side. Those which are funded by the National Institutes of Health in the US are in blue. Those which are supported extensively by the Human Cell Atlas effort are in red. And of course, it's not a complete list because that list keeps on changing over time. Some funding runs out, new funding comes into existence. New unfunded efforts make a huge difference also. And so it's not a matter of funding, it's a matter of how many people really believe that we can do this together. And oftentimes we will be able to do a lot alone, just human intelligence, but I think we will have to bring in massively artificial intelligence to really understand the structure and function of the human body across scales, across temporal scales and spatial scales, and there are many of them. And so here on the right-hand side, you have organs that some of these consortia care about. Many of them care about lung because this all started during COVID. And so many were interested to understand how vaccines can be developed, what long-term implications COVID might have. But there are many other organs which are well-funded and near and dear to many people's hearts. And ultimately, no single organ can do it alone, right? You need oxygen from the lung, you need nutrients from the intestine, you need the brain, you need all of them. There are many, many vital organs, and that's also where research has been focusing on. So some of you might be familiar with the Human Reference Atlas. Others might have never heard of it. I'm actually curious to see how many of you are familiar. If you could give me a show of hands. So if you have heard about it before, could you raise your hand? And I guess there is a digital way to do it. Let's see. Maybe there's nobody so far. There's one. Chris Wood, thank you for, and the LIBRE Hub. So let me just give you a brief introduction. So the Human Reference Atlas is really an effort to agree on terminology, but also to ultimately get us a common coordinate framework that allows us to semantically and spatially and demographically capture the anatomical structures, cell types, and biomarkers in the human body. And so we work with so-called ASCT+B tables, which have a tautology of anatomical structures here for kidney. Every single anatomical structure is linked to an Uberon term. If there is no Uberon term yet, we have temp IDs, and right now, more than 1,000 terms need to be added to Uberon and CL so that healthy human is covered properly in these existing ontologies. So that's another part of this community effort to massively change the ontologies that exist right now. Then you have a tautology of cell types, so that's the ISE, with cell being the top root node, and then going down, becoming more and more specialized cell types. And then there are biomarkers such as genes, proteins, lipids, metabolites, also proteoforms and others, which are used in different assay types to characterize cell types. And it's very interesting for me to understand also what kind of cell types you can identify using these different types of biomarkers and how that relates to each other. If you have two butterfly tissue sections, which should have similar cell type populations, but maybe because one is submitted to single-cell RNA-seq and the other one is submitted to Codex, you have very different cell types that you actually identify. And so ultimately, this all needs to be multiscale. So from the meter-sized human body, male and female, down to the biomolecular level, which is 1 nanometer. So it's 10 raised to the power of 10 zooms, which people are not very good at making in their mental mind. So we have all kinds of different interfaces to help support that kind of zoom. And similarly, also, there are temporal scales when you look at functions. So some processes unfold in less than a second and others unfold over your lifetime. So that's also almost 10 raised to the power of 10. zooms right there for the temporal scale. And so ultimately you can have a 3D plot of all the temporal scales times the spatial scales times the many different physiological systems, and you get to see what kind of structures and functions are relevant in these different cells. So again, the initial 7 years of this effort have been on trying to agree on terminology. What is an atlas? What is a map? What is a cell type annotation tool? What is a crosswalk? What is a common coordinate framework? And then also have examples of this atlas in the making. And so there is a process that is how to construct the atlas, and then there's another process which is how to use the atlas. So let's first look at the construction. So we have many human experts writing in Google spreadsheets what anatomical structures are relevant for their favorite organ, which cell types exist in which anatomical structures, and also what biomarkers they commonly use to identify these cell types. So these ASCT+B tables exist now for 34 organs. They can be accessed via an API. They are part of a knowledge graph. And they are used widely in different portals also to start to harmonize data sets. And of course, they're also used to update these existing ontologies. Then you have 3D reference objects, which oftentimes have a lot of inner structures. So you see the heart with a lot of anatomical structures inside of the heart. And each part of these anatomical structures has different cell type populations. So here you have 2 anatomical structures highlighted, and then different types of cell types and the number of cells for that cell types, or here, percentage. We also have functional tissue units, which are alveoli in the lung or glomeruli in the kidney, and they are currently only available in 2D. We are absolutely interested to get them modeled in 3D, but the data has been hard to come by because these structures are 300 micrometers, around that size. And so it's tricky to get them via imaging technologies. And of course, we ultimately want single cell data, and it's just very expensive to get that single cell data in 3D in that size. But if you have links pointers, I would welcome those. Then we have a lot of machine learning algorithms which do spatial segmentation of the imaging data. We do proteomic imaging. We have so-called organ mapping antibody panels, which are standardized antibody sets that can be used to understand healthy human tissue. And of course, we have a lot of cell type annotations that can be used to annotate cells in single cell or single nucleus RNA-seq data. And so for the lung, for instance, there are 7 different cell type annotation tools, and probably more by now, that have been used, and they all have slightly different annotations for the very same data set. And so we are trying to understand which data set is best for which assay type, for which organ, for atlas construction. We also went into the biomed literature, especially PubMed, because it's a public available data set. And we have the Human Reference Atlas digital objects, including the ASCT+B tables and the ASCTs and Bs inside of that. We have these OMAPs that are other digital objects. We have creators of these tables and reviewers. And we can compare that with the publications in PubMed, but also the projects that are funded and the funders. So we were interested to understand who's funding all that atlasing work. But of course, each publication also has authors, and we are drawing from these authors that are experts on certain organs to recruit reviewers for the ASCT+B tables. And if you would like to serve as a reviewer for the Human Reference Atlas, please let me know. Here you see some of the anatomical structures that are modeled in 3D using the Visible Human project data by the National Library of Medicine. And so these 3D organs have, again, very detailed inner structures. So they are not just the outer surface of the organ, but there are a lot of inner structure. And they are then used to register tissue blocks into it. So for instance, here you have the kidney, see many different parts of it, but then also combined as a reference object. And then if you do a kidney biopsy here, you would ultimately register that tissue in here. This way we know if it was taken from the upper lobe or the lower lobe of the kidney, if it came out of the cortex or from other anatomical structures. Recently, we had a very interesting collaboration with dentists, and they modeled many different anatomical structures in the mouth, and have also contributed single cell data for the mouth. And apparently that's very important also for not just mouth health, but also gut health and ultimately brain health and the health of many other organs. Here you see a screenfall of functional tissue units, and there are more now, including for the mouth. And for the skin, we have the hair follicle for the skin now also in this set. And you get to see that these functional tissue units are at different levels of size. So here you have 0.1 millimeter, 0.25, and then up to 1 millimeter for the nephron tubule. The hair follicle is also in that range. And so you get to see them for the very first time all together. And those are the functional tissue units where you have teams of cells work together very efficiently to deliver critical functions that is absolutely necessary for you to live. And so we have been using these functional tissue units to understand what changes as we age, what changes as disease comes in, ultimately also what changes across sex or ethnicities and other demographic groups. Again, all of this data can be accessed online. So here, if you, for instance, go to the KG Explorer and you search for your favorite organ, if it is lung, you will get to see this screenfall. You can then download all these different types of objects and can use them in your teaching or in your research or to, again, do commercial adventures. This is a video which we just tried and it didn't really work due to the bandwidth we might have here. But Tobias has a link to the YouTube video, so you might like to look at it. Basically, it guides you through the many different scales from 1 meter to 1 nanometer of the human body, and then back again, kind of like the Power of 10 movie that some of you might remember. And it's this multi-scale voyage, but this time you have all the data and all the codes that was used to create this video available to you. So again, you could recreate the video if you wanted to do this, but also if certain scales are more relevant for your work or for your teaching, you can go in here and just show this magic voyage. And we hope to inspire future clinicians, but also anatomists, pathologists, and other researchers that work in this range to embrace the complexity and the multi-scale nature of the human body, and to ultimately also help us get not just the structure, but also function digitized. So again, if you go to this link here, you get a full provenance record of all the data and all the codes that was used to create this movie. And I think that's the first movie where you have something like this. That is more on the autistic 10 raised to the power of 10 side. All right, so using the HRA. So let's assume we now have an atlas. Then what you would like to do is to register a new tissue block here, for instance, in the colon, large intestine, and to get a prediction of what cell type populations are normal for a certain donor demographic. So let's say you are interested in male and a certain age range, and maybe also a certain ethnicity. So thanks to the fact that now we have quite a bit of data registered, we can then start predicting cell type populations. So number of cell per cell type, but also biomarker expression values. What you can also do is after you cut that tissue block, oftentimes it's sectioned up with a microtome in 2D sections. We now have code that is more and more widely used across the HRA community to then do a 3D reconstruction. So this is by the GE skin team. And all of that code is licensed in a way that anyone can use it. You might also take one of these tissue sections and run single cell or single nucleus on a seq assays over it. Or you might run proteomic imaging or H&E stains. H&E is very critical for us because we can use code to segment out functional tissue units and then look inside of the functional tissue units and get the cell type populations for just those functional tissue units, especially if you have, for instance, codex data and matching H&E data, then you can absolutely do that. And again, also that code is all available. And then given transcriptomic or proteomic imaging data, you can then also use the cell by gene matrices to compare it with the ASCT+B tables and with the atlas and start predicting where that tissue might have come from in the human body. So you have both sides. So you can go from tissues that you cut and predict cell type populations, or you have cell type populations and you start predicting where in the human body that might have come from. And right now, where it might have come from are corridors, because oftentimes a tissue block might write on, if you wish, two different anatomical structure sections. Maybe it overlaps 30% here, 70% here. So it basically gets to have cell type populations from both of those. In some rare cases, it was cut from a part where three structures come together. Then you just get one tissue block predicted. But oftentimes you would have corridors that are returned. And again, there are a number of user interfaces that now makes that possible without any kind of coding abilities. And it's just visual user interfaces for that. So ultimately, the atlas is also used to not just take a tissue block and predict cell by biomarker matrices, or take cell by biomarker matrices and create and predict these corridors, but it's also used to compare, for instance, here, healthy lung to diseased BPD lung. And we are using the cell distance explorer here, which gets you cell distances from different types of cells to vasculature, because we are trying to see if we can use the vasculature as a latitude-longitude system for the human body. The Cartesian coordinate system that is currently used to register tissue only goes so far, because we all have quite different body shapes and sizes, and also the inner organs might touch or not. So a vasculature-based common coordinate framework seems to be much more desirable and accurate ultimately, but we are still in the process of getting the right data for that. But what you see here is that you absolutely have differences in diseased versus healthy cell type populations. Among others, new cell types might come in if a tumor cell is detected, for instance. And I will have more examples of that later. Down here, under D, you see work by John Hickey, who used to be at Stanford University, now has his own team at Duke University. And he's very interested to automatically identify cell types, cell neighborhoods, cell communities, and ultimately functional tissue units. So it's kind of a nested approach of grouping cells into neighborhoods that are meaningful, functionally meaningful, or are always or oftentimes seen together. And ultimately that is changing as disease comes in or as we age. And so this hierarchically nested set of functional tissue units has been super valuable for tracking disease states, but also helping us understand what changes as we age or disease comes in. And so here, for those of you which are interested in lung, you have Gloria Pryhuber as the lead contact, but many others contributed. And you have the human body, you have the lung, you have the many, many anatomical structures in the lung, you have cell types that are in these lung anatomical structures, and you have the biomarkers, and you have also a report of how many anatomical structures, how many total cell types, how many total biomarkers exist. And you have that for any of the 34 organs, and you can explore that in your own time in the Explorer. But you can ultimately also just visually examine it. And that has been very helpful for harmonizing the naming and terminology, but also helping people benefit from this effort that we have been leading over the last 5 years now. So here for the lung, heart, and kidney, you have these white blocks, which are tissues that have been registered. So these are the areas where tissue data exists. Sometimes some teams might take a biopsy always from the same place in the human kidney. They use ultrasound-guided biopsy extraction to make sure that it is in that range, even though different human beings have different size of kidneys, also different position of the kidney and shapes, of course, also. So there is a combination of what human surgeons can do and what the current registration user interface allows you to do. And again, ultimately that registration is needed so that we know where the tissue came from, so that in the future, cell type populations can be predicted for the right area in the human body. And then you have this exploration user interface that allows you to explore more than 6,000 tissue data sets, and many of them are single cell. So if you are interested in running meta studies or just seeing who has what kind of data using which assay types, that would be your place to go. We also, for those of you which have a VR headset device, have a so-called HRA organ gallery. And in that organ gallery, you can face in real life size the male and female reference bodies. And then you can zoom into, again, your favorite organ here. We have lung. And you can then also zoom into different elevator levels, which correspond to these different 10x zoom levels, and get to see different organs at different scales of resolution. So Andreas Bückler, he is part of our team here at Indiana University. He would be happy to come back and give you a demo of that if this is relevant for your work. And he also led the HRA POP effort, which is the cell type populations across different anatomical structures in the human body. So here you have quite a few male and female specific organs. So we have the reproductive system organs in here, which is also a first for these atlases. Oftentimes there's only one atlas and it's for male, and oftentimes it doesn't have some of the reproductive organs. So we are very proud to have that covered. And then if you have your favorite organ, for instance, small intestine, you would have 1, 2, 3, 4, 5, 6, 7, 8 different anatomical structures and the cell type populations that are commonly found in them for female on the right and male on the left here. And then again, this is all available programmatically also. So if you want to compare the cell type populations that you see in these anatomical structures, maybe for diseased tissue, to the healthy normal ones, that would be one way of doing it. Here you see the FTU Explorer, where you have interactive FTUs, where you can hover over these cell types and they actually highlight them. So if you go to the app itself, you will be able to do this, or you hover over these cell types listed over here, and they will then also highlight the corresponding cell types in the here bronchus submucosal gland. And then you get to see the number of cells of that type in a certain data set. And you also get to see here the gene expression markers, and you can hover over these size-coded symbols to get more information. And again, you can do this for 27 functional tissue units now, and for some we are still adding more data, but it's all set up so that new data sets can be added easily. Here you have the illustration for the alveolus, and again, different cell types and also different biomarker expression values. The tool that I think you might be most interested in trying out on your own data is the Cell Distance Explorer. So the app is right down here. Tobias, if you want to put that link into the chat. If you have data of single cell types, for instance, or boundary markers or disease markers, and you have an XY for those, and if you have 3D data, you can also provide a third dimension, and you have a name, which could be a cluster name or a cell type name or a damage marker name or a protein marker name. So this is very flexible, this tool. Then you can upload it and you can get to see your data in very new different ways. So again, we are typically interested to understand distances of different cell types to vasculature. So all the visualizations you get to see next are doing that. But other teams have now started to explore distances of certain cell types to nervous system or to certain immune cells or to certain damage markers or to certain tumors. And so it would be really neat to see what you will do with this tool. And again, it's freely available. Anyone can use it, and you don't need us any longer. It's for you to use. And there's a recent study out where you actually have 14 studies with 12 tissue types and a total of 47 million cells, all run through this cell distance explorer, so that we get to understand what cell types are shared across organs, but also what's specific, for instance, to the intestine as you go through the small intestine and then the large intestine. What kind of cell type populations and cell neighborhood distances do you encounter as you go from basically the beginning of the intestine to the very end? And these kind of analysis, again, are super valuable for atlas construction, and they might also be useful for your work. So here you have the different data sets that are in that paper I just mentioned, the 12 tissue types from different teams. Some of them have disease included. You also have other visualizations of papers that have been published earlier. And so here, for instance, you would have colorectal cancer. And as stages of cancer progress, these distance distributions change rapidly. And they are really a good marker for different disease stages. And here you have different immune states also, if you're interested in immune cells. And so again, these distance distributions to vasculature, they are on our end used to plot all these anatomical structures, cell types as a so-called butterfly visualization. This is actually printed in large, like 3 by 3 meter, and you can put yourself in front of it and take a selfie. But then, of course, you also see the anatomical structures, the functional tissue units here, which we are focusing in on, because those are the teams of cells that are working to get us the function we need. And again, it's multi-scale, and you also get to see how the vasculature might be used ultimately at the macro scale, mesoscale, and microscale to get us a zip code system, basically. So in the US, each zip code is a lat longitude coordinate. And we need something like this for the human body. There is a video that explains how that might work, because it's not easy to understand how a combination of cell distances, cell type populations, but also diameters and branching angles and other morphological features might be used to get us basically a postal system for the human body. But again, when we tried it before you all came on, the videos didn't run, so maybe Tobias just put the YouTube link into the chat, and I'm not going to try this here because it's no fun to watch it if it just is like in bits and pieces. So this human reference atlas really benefits from the human cell atlas, which is older and has been super supportive in getting the community together to work on this multi-scale reference atlas. And here you just see the HubMap team and the team we have locally here at Indiana University. But ultimately, there are now more than 300 experts DOI'd with their ORCID IDs connected to the Human Reference Atlas, and there are many more also using it and coming into the HRA working group that we have every month. And so maybe we can also share more information on that working group if you're interested. And again, all of this wouldn't be possible without funding, because single cell studies are still very expensive. You need highest quality data to create an atlas. And you need also the expertise to wrangle that data, to have a data portal, to ultimately analyze that data, model it, and visualize it in a way so that insights from these initial studies can also be used to optimize assay types and protocols and OMAPs and single cell type annotation tools, etc. So it's a huge community effort. So you see me here, but there are really 300 people on this Zoom call presenting this to you. One other piece that you might be interested in when we talk about the multiscale human body is the CIFAR Macmillan funded project, which again just met thanks to Tobias in Chile, which again I missed. But I think Tobias is your go-to person there. He has organized it all, and he also was able to attend together with many other experts from around the globe. So thank you for having us all there. And this particular CIFAR program also has an open call right now, and anyone from anywhere on this planet can apply by December 17th to come to the Multiscale Human Summer School, which will be in May in Vancouver. Everything is fully paid for. There are always very nice venues and lots of amazing experts coming together. And of course, we would love to have students from all continents. So please do share this widely with your students and others that you know around the globe. Again, everybody's welcome, and we would love to have you all send your students. So it's mostly for students and postdocs, not necessarily faculty, maybe junior faculty. And if you want to learn more about this effort, there's another YouTube video which explains how all this data and code might be used to create digital twins, which come into existence when we are born and then are with us as we go through life. And whenever there is a new procedure that's needed, it would be first tested on the digital twin before it's applied to your biological body. So I think there are lots of major ambitions in this program, and you might enjoy it. So maybe I pause here to see if there are questions that I can answer. And then there's a second part on data visualizations, which maybe I can do in 5 minutes. But let's see if there are any questions so far.
This was really great. If there are questions in Spanish, I can also help to translate. Si hay preguntas en español, también puedo traducir.
The same, well, también en portugués podemos ir. Traducir.
And I see that Tobias really posted a lot of the links. Thank you so much for doing this. I know this is a lot to take in because it's 7 years of work, of very intensive work. But I see Andres Kameda has a question.
Hi, Katy. Very, very impressive. I am absolutely shocked by this presentation. Thank you very much for sharing all that with us. Mine is kind of, first of all, from that shock, maybe a general question about, as a network of bioimaging scientists, what would you advise us to do with how can we spread the word, and besides this kind of seminars, what would you recommend us to think about concrete actions to make this more available to our community?
So I hope that the links that you have in chat empower you to share it with your students. I think in the very beginning, I also posted the link to the Visible Human MOOC, which is a massively open online course where lots of us try to communicate how we could build an atlas together and how it could be used. So please do share that. Everything is free and open. And we worked very hard to make it that way. We had some collaborators which needed 3 years with lawyers to make their data and code available publicly. That has happened, and it's now available. Ultimately, we need expertise from organ experts and single cell experts, pathologists, clinicians. I myself, I'm an engineer. I serve the needs of others. I can scale up data visualizations and user interfaces and knowledge graphs, but I am not trained in surgical procedures or trained to work in a wet lab environment. So I think this needs to be a team effort. And you always have that quote, if you want to go fast, go alone. And if you want to go far, go together. And we absolutely want to go far. And so we are trying to help everybody understand that this has been accomplished so far, but we have much more ambitions. Like recently, the National Institutes of Health funded a new project on the human physiome, where you really look at function. So everything I've told you so far is all about structure of the human body. It doesn't yet look at the many feedback cycles that keep you alive. Many of them are negative, to just keep something in stasis, right? Calcium, glucose, all of that. And then some of them are positive, to give birth, to vomit, to other things that oftentimes have a disruptive ending. And so we are trying to capture all of those feedback cycles, but that's a totally different project and a different effort. But we do benefit from the Human Reference Atlas because we already have a lot of anatomical structures digitally available. We have many of them available in 3D, and life does unfold in 3D. And so again, if you are interested to use any of what I presented, feel free to do that. I always like to hear back how you used it, but you don't have to do that. If you would like to contribute, contact me also. We always need reviews for organs. Currently, we need a heart expert. So the heart expert we have been working on, he changed positions. So now we need to find another lead organ expert. So there are always changes, and we would love this to be an atlas that's collectively created and collectively used, even though it started maybe out of Europe and US. Ultimately, it should be a worldwide atlas.
Can I follow up on
Thank you.
Andre's question? Maybe Andre also has a follow-up, but just from my experience, so there, as I know already, there are several sort of young researcher events from the Human Reference Atlas, and you're trying to get people involved. What are the ways in that initiative specifically to get involved on different scales, other than being, you know, the expert of an organ, which is quite a heavy burden to carry, I guess?
Yeah, so again, we regularly send out the digital objects to organ experts. And so you would get to see ASCT+B tables, you would get to see 3D reference organs, you would get to see FTUs. And at any moment in time, you can also just send us comments. So on the humanatlas.io website, there is a general email address that we monitor. So if one of those digital objects could be improved, please let us know. There is also a lot of effort to engage the next generation of researchers. So NIH, in collaboration with the Chan Zuckerberg Foundation, have been hosting junior atlas builder meetings, and those have happened 4 times. In the last year, it was virtual because of all kinds of things that are happening in the US right now. But I can put the link to those into the chat because there are there is good information on what is done. It's more like a career development workshop, but again, it's held in a way so that anyone can attend it. And here is the agenda. So we would love to have more global participation. This should not be just confined to Europe and the US. So I put the agenda for the most recent Junior Investigator Atlas Builder meeting, which was virtual, into the chat. And maybe in the future, Tobias, I can also just share them with you and you can share them further. And over time, we will create more and more bridges between these efforts. So maybe I present a few more slides because I wanted you to also see that we do a lot of data visualization, and that's maybe also another way to connect. I teach every spring 130 students how to analyze data temporally, spatially, geospatially, and also in networks. And so they have to analyze these data sets to answer when, where, what, and with whom questions. But they also then, in the second part of the project, get to work with real-world clients. And so for these real-world client projects, we are always interested to learn about data sets and experts that have good questions, but maybe no funding to analyze and visualize that data set. And those projects are perfect for the students because they have a meaningful data set in question. They work with a leading expert or with a PhD student who is smart enough to use that opportunity to jumpstart their PhD work. And we have clients around the globe, which ultimately our students will also have when they go into their work job environment. So let me just show you a few of these visualizations to get you interested. And then we can have another set of Q&A. So data visualizations, I strongly believe that we need them to understand more globally and long-term what is happening on our planet, be it in the job market, be it in science, be it in technology developments. And data visualizations really can help there. And oftentimes you also have a multi-scale structure of your data. You might have personal data from your Fitbits or from your bank accounts. You might have professional data from your job. And of course, keep those separate, please, and don't give your personal data to many people. So all of this is part of it. But then you can start rendering data statically. So we had a map of science exhibit running from 2005 to 2014. And it made it to Mexico during that time. And we actually have an ambassador in Mexico, which I could reconnect with. And she has been good in bringing this exhibit as a poster version to many different places. But then there are also different atlas books, which basically explain how why you would want to map science, how you can make your own maps, but also how you can use predictive models to ultimately model and map desirable futures. And oftentimes people want to first test run a future before they sign a check. So they really want to use existing data to understand what happens if I do fund X or fund something else, or what happens if I make this kind of economic decision. And again, all these maps of job markets and science landscapes and IP holdings, etc., are super helpful for understanding trends and clusters and outliers. And so this entire exhibit is available under scimaps.org maps. And that was the initial 10 years of the exhibit. And we brought the exhibit to many, many different places, oftentimes as frame maps, also in larger format. We have this interactive panel display here. And it was really nice to then go to these places and also kind of do curatorial walkthroughs. And maybe at some point, the exhibit could also come to Santiago, and I get to see Santiago. I would love that. But it's quite an effort. So maybe first get tenure and then do an exhibit. So it's really a lot of effort. Then the next decade of the exhibit is all on interactive data visualization. So using this big touch panel display, Liesl and Todd and myself have been curating this for 10 years. And the book just came out. I just got my own copy, but I think it's now available online. And so this was 2015 to 2024, surviving COVID, where we couldn't have the exhibit on display anywhere. That was hard, but now you have a 4D interactive data visualization. So it's not printed static maps that you hang on the wall, but it's maps where you can look for a certain keyword. You can zoom in, you can get details on demand. So it's a very different way of interacting. And in many cases, you get inspired to see your own data in this way, and then we can connect you to the Macroscope makers, which made these interactive data visualizations, and you can render your own data in similar ways. So it's also spreading the word about how you can take interactive data feeds and render them in meaningful ways. And so again, here we have the exhibit at the University of Michigan Library, and you see the panel, but also some of the maps on display. And then much had to happen online during COVID, and we did a lot of interviews with map makers, and they are all online available. We have more and more interactive interfaces, but then also often with masks. So it was really hard to have an exhibit, and as you know, many science museums had to close their doors and had to let people go, and it was just really hard for science communication during that time, and I think we still have not totally recovered from that. So I think it's really important that science goes out of the ivory towers and reaches general audiences, and the exhibit has been magic doing that. And so here you have the panel, you can click through these different maps. There's a smelly map, which shows you different smells in here, London, but also shows you where in London people are joyful or fearful or sad, where they have trust. And I think that something like this is super valuable for urban designers also, because you want people to not have fear in your city. And there are many, many maps like this for other cities. I'm not sure there is one for a city in Latin America. So you might like to check it out. All of these maps are available online. This is a set of mega regions based on commuting patterns. I'm running out of time, but happy to come back and explain it. News co-occurrence, who is talking about what under other country in the news. US typically doesn't talk about other countries much, but many of the African countries talk about many other countries outside. So it's really super interesting to see that dynamic unfold. Job market, which jobs have low salary and low growth? Maybe you don't want to go there. And which ones are high growth and high salaries also in the future when AI and robotics and automation moves in. So really interesting set of analysis, and they had to create a lot of ontologies and taxonomies to just harmonize so many different data sets to have these kind of insights. So there's a lot of data wrangling behind each data visualization. And so here you see all 40 of these data visualizations, and I hope you get a chance to explore some of those. We currently have a set of book reading tour events. So this actually today I count as a Santiago, Chile, Latin American bioimaging event, a virtual one. So thank you again for having us on. We did some local ones in Bloomington, Indiana, where I am right now. But we also tried to cover Africa and many other places so that the word spreads that you can do render data intelligently into actionable knowledge. And so very excitingly, we just committed to another decade of the exhibit, and maybe that's the last decade. I think 30 years is enough for an exhibit. But this will go on till 2034. If you are interested in intelligence, which you should be, then you might also be interested in interactions between the human microbiome and us humans. We absolutely need the microbiome to live. We're also interested to see how trees and fungi interact, or bacteria and viruses. And of course, there will be submissions on how we interact with AI. And so we take intelligences very broadly. There are different types of intelligences. There is collaboration and coordination going on, and some cooperation and collaborate and competition is oftentimes good if it's in not too bad, because you really don't want currently the world is really in an interesting state. I think it's better to collaborate and coordinate. Ultimately, we would like the exhibit to inspire discussion about existing and future sensors and actuators, also for us. I think there are some in the making, especially also for those which have lost some of their sensors and actuators, human ones, but also memory and reasoning, exploration and communication, but also shared goals and desirable futures again. And so as part of an event that is coming up this weekend, actually, we will also discuss the sentient architecture that we have hang in LABI Hall here in the building. And that structure has a lot of sensors and a lot of actuators, and it really is interacting with you all. And you can absolutely visualize what these sensors and actuators are doing. And so this weekend, there will be a 24-hour around the globe event where we have experts from around the globe come in and discuss how we can visualize intelligences, how they interact with each other, what they do, what capabilities they have, where they fall short. And of course, that's really important also for understanding what AI can and cannot do. But again, it's not just AI. And so we open at 12 o'clock Eastern Standard Time. And then for 24 hours, feel free to come in. This is the link. Maybe Tobias can put that into the chat. And we have done this before. We started during COVID with this on science maps, and we had an entire session on the Human Reference Atlas. We had one on the interactive data visualizations. And then last year, we had one on multiscale human data and code. And so all of this was recorded, so you can also go back to those. But this weekend live, you would get to see me for many hours, and then also my collaborators. And we are going to do it. And it's always exciting and exhausting, but I hope you can participate. And so here are the co-curators and also our advisory board. And again, there are more than 250 authors which have contributed maps and macroscopes to the exhibit. And that's the end of it. Thanks so much for being here. And I would be happy to answer more questions. I do have a few more minutes. But thanks also just for having me here. And I would love to connect more. Also, thanks to and via Tobias.
And the fantastic LABI crew who was very willing to jump onto this seminar and new format. So thank you very much for an amazing presentation. As always, very inspiring. So thanks for being available for a few more minutes of question. I definitely want to make use of it. So please, if anyone would like to ask anything, this is your opportunity. Since you connected live, the people that will just watch the video will only be able to write messages later.
Thank you, Ola. I apologize. I have to leave soon to another. Will be
Good to see you, Andres.
Yeah.
Yeah, it's always a lot to take in. I mean, I've had the pleasure now through the being part of the CIFAR program to interact a little bit more with, well, you, but also other members who are in charge of some of these data initiatives. And it's, if you don't already work with it, it's just a little bit difficult to get in, I think, and understanding how you can use it there. Especially, there's a lot of now single cell and spatial transcriptomics, which is generally a little bit too expensive for us in Latin America. There are some exceptions. But these can still be really fantastic resources to enrich our and inform our own experiments. But yeah, I'm also personally like looking into how to do that if anyone is willing to interested to discuss it or collaborate. That's great. But yeah, there was a lot of interesting, I think just keep exploring all these very cool links that we've seen, the maps, the macroscopes. There's a lot of a lot of things to explore still. But if you have any thoughts or questions now, please do go ahead and ask.
Just
Don't
to
be afraid.
translate, there is a comment on the chat that says ver es comprender. That means seeing is understanding. So
Oh,
you get
I
it.
see. Thank
Yes,
you.
very good. Yeah, or seeing is believing, as we often say in English, right?
And you can lead people astray, right? By picking the wrong data or the data you wanted to see and visualizing this in high quality so that it looks like it's very professionally done. And then others taking it for face value. I think that's also a power you have. Any map maker has that power. And so we have to tell our students that we will make them powerful, but then they have to use that power wisely.
Well, and in contrary, I think having these initiatives is really valuable because you can double check claims with available data sets, right? So
yeah. And that's what we are also doing with the Human Reference Atlas, right? So we have the human-generated data, the ASCT plus B tables, then we have the experimental data, which we can compare with the cell types that they have listed in the ASCT plus B tables. We should see them also in the experimental data. And then, of course, there's also the massive set of scholarly publications, which you can now, thanks to the large language models, mine and analyze, and then also compare to the atlas. And I think this cross-checking between what's in human minds, what's in the literature, and what's in the experimental data, then hopefully gets you a more and more comprehensive and accurate atlas of the human body.
That's great. Just a comment for the rest of the people. You will receive, of course, then you can access this recording into the YouTube channels. You will receive an email also with all the links that we have shared on the chat, so you can have it all together later. And we can place it on our website and share it, of course, by email. With that, maybe we close for the rest of the people. So we have on time.
Thank you so much.
Thank you very much, Tobias,
Thank
for
you.
leading this.
Yeah,
Katy,
thanks
for
for
your
having me.
impressive
It's great
presentation
to
and all the
connect.
work that you are doing. It's impressive. Thank you
Yeah,
very much.
and Tobias, we will see each other in Vancouver, right?
Yes, I'm looking forward to that. Exactly.
So there is one strong connection because we see each other twice each year. So yeah.
Thank you very much for taking the time to share this with us, Katy. That
Yeah,
was really
no, thank you
interesting.
for having us. We really want these bridges, especially right now. Okay, bye.